Okay, so now moving on to our third macromolecule of life, and that's going to be proteins. So while carbohydrates and lipids really help provide energy and structural components, proteins do nearly everything else. So think about things like enzymes, antibodies, hormones, muscle fibers, you name it, proteins are going to do it. So what are proteins? They're polymers that are made up of amino acids. So amino acids are going to be their monomers, what we're going to link together to make this macromolecule. And they have a wide range of functions. So it's going to include things like support, if we're talking about collagen and connective tissue, metabolism, so when they act as enzymes to speed up reactions, transport, so things like hemoglobin carrying oxygen, also defense, if we're talking about antibodies that are fighting pathogens, regulation, if we're talking about hormones like insulin, and even motion, if we're talking about muscle proteins like actin and myosin. So each protein is built from a unique sequence of amino acids, which is going to determine its final shape and function. So all amino acids have shared the same core structure. They're going to have a central carbon atom, which is called the alpha carbon, an amino group, and an acidic group, which is a carboxyl group. So that's why it's called an amino acid. It's also going to have a hydrogen atom. Each amino acid has a unique R group or side chain, and that's going to determine the identity and the property of the amino acid because there are 20 different amino acids, each with a distinct R group. So these small differences are going to create a huge diversity of proteins. So this is a good image over here. Here's the general structure of an amino acid. We have an amine group, and then our carboxyl group, which is an acidic group. That's why it's called an amino acid. And we're going to have a hydrogen atom, of which is bound to a central carbon atom. So all amino acids have that same exact structure. What makes it one of the 20 different amino acids is going to be dependent on what the R group itself is. So they're showing you over here to the right 4 different potential R groups on an amino acid, which is going to determine which of the 4 shown here amino acids it actually is. So when 2 amino acids are joined together by that dehydration reaction that we talked about, they form a peptide bond. And this is just a covalent link between the amine group of one amino acid and the carboxylic group of the next. So a peptide is just a short chain of amino acids. And if we have a polypeptide, that's a longer chain of amino acids, essentially what we refer to as the primary structure of a protein. And then these chains can fold into complex shapes, and that's going to determine how the protein functions. So it's just showing you here a dehydration reaction going in this direction to link amino acids together. The bonds between amino acids are what we specifically refer to as a peptide bond. If we want to break apart the amino acids, we have to go through a hydrolysis reaction, which is the same for any of our macromolecules. So let's talk about the levels of protein structure. We have the primary structure, and this is just the linear sequence of amino acids in the chain. So this sequence is like a code. Even one change can dramatically affect function. For example, I think we'll talk about later sickle cell anemia. If we're talking about the secondary structure, this is just local folding into either alpha helixes or beta pleated sheets. This is going to be stabilized by hydrogen bonds. Tertiary structure is going to be the overall 3-dimensional shape of the protein, and this is going to involve interactions between R groups, including ionic bonds, hydrogen bonds, and hydrophobic interactions. And then we have our quaternary structure. So some proteins are made up of multiple polypeptide chains coming together to form this 3-dimensional structure, and their final shape depends on how those chains are actually going to interact. So hemoglobin, which is a pigment in our erythrocytes or our red blood cells responsible for transporting oxygen, actually has 4 subunits that are working together. So this would represent a quaternary structure. So this is what it's showing you here. This is our primary structure here, which is just a linear sequence of amino acids. So we can see the abbreviations for each amino acid here, and they're just being linked together. When we start folding either into alpha helixes, which is shown next, or beta pleated sheets, this is going to represent the secondary structure. Tertiary structure is going to form when we have an overall three-dimensional shape, but only with one linear sequence of amino acids. If we take more than one linear sequence of amino acids and put it into a three-dimensional shape, that's when we get the quaternary structure, which is shown here. I just want to mention one other thing. So a protein's function depends entirely on its shape. So when proteins lose their shape, they become something called denatured, so meaning that they're not functional anymore. And different things can cause denaturation of a protein. So things like changes in pH, which is why buffers are so important, temperature, salt concentration, all of those things can cause our proteins to lose their three-dimensional shape, and proteins like enzymes for them to stop working correctly. So this is why really high fevers or an acidic blood pH can be so dangerous.